Hello everybody, I welcome you all to this video lecture on Puccinia. It is prepared for the benefit of English medium students. The video lecture is being given by D. Rajasekhar, lecturer in Botany, Srimati Ganganeni Kalyani Government Degree College, Vinikonda, Guntur district. It is included in the syllabus of 1st BSc BJC in the 5th unit, that is fungus, where we have to study different 4 fungi. They are Rhizopus, Penicillium, lichens, and Puccinia. Students generally feel Puccinia a tough topic, but if you comprehend the topic clearly, it would be very easy for you, even very easy to attempt the questions of this topic during the examinations. Puccinia is an obligate parasite which causes rust disease to wheat plants. So far, 4,000 species have been identified. All these species cause rust diseases on different grain plants. In this video lecture, have mainly focused on Puccinia gramineus triticale, which causes rust disease to the wheat plant. It was identified by Puccini and named by Pessoon. Puccinia belongs to the subdivision Basidiomycotina, class Teleomycetes, order Urodinales, family Pucciniaceae. As it completes its life cycle on 2 different host plants, it is also known as heteroecious rust. The first host plant is wheat, that is also called primary host, and second plant is Barberry vulgaris, it is the secondary host. In its life cycle, Puccinia forms 5 different spores, hence the life cycle is very long. So it is called macrocyclic life cycle, and the disease is called macrocyclic rust. Our Indian scientist Karamchand Mehta discovered the life cycle of fungus and suggested the world not to cultivate wheat crops in high altitudes. If we do so, the wind-borne spores of Puccinia easily carried by the wind currents to the lower altitude regions where they attack new crops and new plants so that the disease severity will be increased. By his suggestion, lot of damage to the wheat crop was controlled to the maximum extent. Any disease can be identified by its symptom. Puccinia causes rust symptoms on the plant parts of wheat like leaf blade, leaf sheath, inflorescence spots, awns, and stem, etc. Different Puccinia species cause different rust symptoms. Puccinia gramineus triticale causes pustule-like symptoms on all the parts of plant body. They are generally reddish brown and black in color. Hence, this rust is called red rust or black rust. When Puccinia develops iridospores in the wheat plant, that is manifested in the form of reddish brown rust symptoms on the plant pods. When Puccinia develops teletospores, the so-called change in the spore formation can be manifested on the plant pods in the form of black color rust symptoms. As I told in the beginning slides, it has 2 host plants. One is primary host, that is wheat plant, and the second one is barberry plant. It is also called secondary host. This Puccinia pathogen causes 5 different spores on 2 different host plants. They are stage 0, stage 1, stage 2, stage 3, and stage 4. Stage 0 is also called spermogonial stage or pycnidial stage. Stage 1 is called AECIAL stage. These two stages are generally seen on the barberry plant. And the stage 2, that is urinary stage, stage 3, TELIAL stage, and stage 4, that is basidial stage, are developed on the wheat plant. Generally, the spores which are formed during stage 0 and the spores of stage 4, that is basidial spores, are single nucleated spores, whereas the remaining three middle spores, such as AECIAL spores, uridinyl spores, and teltospores, are binucleated structures. Life cycle of Puccinia on barberry plant. When basidiospores come from the wheat plant, fall on the barberry plant, they germinate and they enter into the barberry plant through stomata or through injuries. They develop vegetatively and then they develop cup-like structures below the upper epidermis of the barberry plant. As we see in the slide, they are like cups or flask. These flask-shaped structures are called pycnidia or spermagonia, where we can see long filaments on the inner surface of pycnidium. They are called spermatiophores or pycnidiophores, which develop spermatia at their apex. They are arranged like chains. When they get detached from the spermatiophores, these spermatia or pycnidial spores come out through the apical pore, where they are trapped by the honey drop. When an insect visits the plant to take the dew, the spermatia or pycnidiospores that are trapped in the honey are attached to its legs or mouth parts. When this insect consults another plant or the same plant, the spermatiospores or pycnidial spores get attached to the female structures, that is trichogyne. But however, this attachment is largely confined when they are of two different strengths, plus and minus. After that, as the common wall between these two structures dissolves, the male nucleus of spermatia or pycnidial spore enters into the female branch, that is trichogyne, where the female and male nucleus undergo many divisions so that dikaryotic mycelium develops, which grows vegetatively in the barberry plant leaf, and they develop a cup-shaped structures below the lower epidermis of the barberry leaf. They are called AECIAL cups and the stage is called AECIAL stage. In the AECIAL cups, we can see long filaments called aesiophores, which develop basipetally arranged AECIAL spores. These AECIAL spores are binucleated, hexagonal or round shaped structures which are arranged like chains. These aesiospores are connected each other by disjunctors or gelatin discs. They are yellow colored spores. During favorable conditions, as gelatin discs disintegrate, these aecial spores get disseminated through wind and fall on the wheat plant, that is the primary host. With this, the life cycle of Puccinia on barberry plant is over. When these aecial spores fall on the wheat plant, they germinate and they enter into the wheat plant leaf either through stomata or through injuries. After entering into the wheat plant leaf, the dikaryotic mycelium grows vegetatively well by taking the nourishment from the leaf cells through hostoria. After a period of vegetative growth, they develop long, unbranched, colorless structures called iridophores. These iridophores develop iridospores at their apex, and this stage is called iridial stage, the first stage on the wheat plants. Iridospores or iridial spores are oval-shaped structures. They are single-celled, binucleated, enclosed by outer spiny exine and inner intern layer, which is smooth. At regular intervals around the spore, we can see 4 germopores. Generally, these iridospores are detached during favorable conditions, and at maturity, they attack new wheat plants or healthy plants. Thus, thousands, lakhs, and millions of spores attack new plants, thus increase the severity of the disease. Hence, iridospores are called repeating spores. They repeat the disease life cycle in different wheat plants. Iridospores are developed only during growing season of the wheat crop. As these iridospores are reddish brown in color, when they are present in the wheat plant, the symptoms on the plant pods appear reddish brown. At the end of growing season, instead of iridospores, new spores are developed on the iridophores. They are called teletospores. At the time of harvesting, during unfavorable conditions, these robust, dormant teletospores are developed. Teletospores are spindle-shaped structures, black in color, bicell. Each cell contains 2 nuclei. Upper cell shows an apical germopore and the lower cell shows germopore on its lateral wall. During unfavorable conditions, teltospores exist in the remnants of the plant body. When favorable conditions onset, these teltospores germinate and develop basidiospores. Basidial stage. It is the third stage of the Puccinia on the wheat plant. In the teltospores, the 2 nuclei in upper cell and lower cell fuse and form diploid nucleus. Meanwhile, the outer wall is broken down so that the inner wall forms a long filament from the apical part of the upper cell and the lateral part of the lower cell. These long filament-like structures are called basidial branches. The diploid nucleus of each cell enters separately into these 2 basidial branches, the upper basidial branch and the peripheral basidial branch. The diploid nucleus in each branch undergoes mitotic divisions and form 4 nuclei. Meanwhile, each branch develops septa so that they form cell-like structures. Each cell-like structure develops a small protuberance that is called sterigmata. Meanwhile, the diploid nucleus in each cell undergoes divisions and form 2 haploid nuclei. One haploid nucleus enters into the sterigmata and the another nucleus remains in the cell. Thus, the stigmata and this haploid nucleus is modified into basidiospore. Basidiospores are uninucleated haploid spores. They are colorless, very light-weighted, and with thin cell walls. They are easily detached from the basidial branches and carried out by the winds, and on falling suitable barberry plants, again they start the new life cycle. With this, the life cycle of Puccinia on wheat plant is completed. If you see the Puccinia life cycle, it is completed in two different hosts. Primary host is wheat and secondary host is the barberry. In two different host plants, it develops five different spores at different stages. They are called stage 0, stage 1, stage 2, stage 3, and stage 4. Stage 0 and stage 1 are developed on the barberry, and stage 2, 3, and 4 developed on the wheat. As it is a very long life cycle, it is called macrocyclic life cycle. Thank you.